All right, we'll go ahead and get started. Thank you everybody for joining us today for our 12th virtual shadowing session with Dr. Giuseppe Legname, who is a doctor of philosophy in Italy. So welcome, Dr. Giuseppe.
So good morning, everyone. And here is just past 7 o'clock in the evening.
I'm sorry, your sound is a little muffled. Is there?
Can you hear me now? Better?
Yes, we can hear you slightly. Is there? There we go. I think that's it.
Yeah?
Yes, perfect.
Is it okay? Okay, so thank you for being here. I'd like to give you an overview about prions, and of course, I will then divide the talk in two parts. A general part, just to give you basic.
Okay, we can no longer hear you. I think it might be the microphone is underneath. I don't know if there's any way to elevate.
The microphone now?
Yeah, that's perfect. Like that, it works. But when you put it down, we can't hear you anymore.
Okay. Okay, so just let me know if something doesn't work. Okay. Okay. So basically, just a very brief introduction. I work in SISSA. It's a university, as a specialized university in the areas of neuroscience, mathematics, and physics. We specialize in basically post, PhD students. We don't have master courses or bachelor courses, but we have only PhD courses. So let me start then. So as you all know, in our lonely planet, we are not that lonely because the population is growing steadily and has been growing steadily for the last 70 years, and it projected to grow even more in the next 30 years. And with that, of course, there will be a prevalence of some different age bracket. So as you can see from in the 1950, the population was pretty young overall. But now this quote, this slice is shrinking, and it's growing the slice of people that are 65 and over. And it's projected in the next 30 years to grow even more. With this, basically, we all know with aging, there are some diseases that are related to aging. And here I just mentioned a few. The most common one are heart disease and cancer. But also is growing the incidence of neurodegenerative diseases such as Alzheimer's disease. Alzheimer's disease is the most common form of neurodegenerative diseases. And the global impact of Alzheimer's disease and in general dementia is pretty high, it's pretty steep, as you can see here from these figures. And basically, just to mention a few items here is that one individual every 3 seconds is diagnosed with dementia, one form of dementia around the world. And the impact to the health system of this disease is pretty high. As you can see from the figures, in 2018, it surpasses the gross product of US, and basically is now around $1 trillion per year, and is projected to double in just 30 years. So as you can see also from this map here, the continent or the countries that are more affected by this form of disease are the Western world, but also Asia is pretty much involved in this. So, what are the causes of neurodegenerative diseases? Well, here I listed the two most common forms of disease, which is Alzheimer and Parkinson's disease, and together side by side with Prion diseases. So this disease basically are mostly sporadic, which means that we don't know exactly the cause of this disease. There are a certain percentage of this disease which is very low, usually 1 to 10%, which has a genetic basis, and basically there are genes that are involved in the onset of this disease. But what renders peculiar prion diseases is the fact that they can also be infectious. And of course, for those of you that are not familiar with prion disease, this is kind of uncommon, and it's shared with other, unfortunately now we are living in a pandemic where viruses like the SARS-CoV-2, which has a nucleic acid basic inheritance principle, and but in the case of prion disease, we will see that prions are a different kind of pathogen, which the only thing they share with viruses is the fact that they can be infectious. So, are the most common forms of the dementia are also infectious? We're working on it, and basically there are some mechanisms that are shared with prions, but we don't have data that support the infectivity of this common form of dementia. Other things that are common between among these different diseases is the fact that they start at a certain region of the brain, and then during the evolution of the disease, they spread throughout the brain, and eventually in the last stage of the disease, basically the terminal stage of the disease, they cover all the central nervous system. And here, for example, we have Alzheimer's disease with the amyloid plaques. We have also Alzheimer's disease here with neurofibrillary tangle made of another protein called tau. And here we have also alpha-synuclein in Parkinson's disease and TDP-43 in amyotrophic lateral sclerosis. So prion disease in human and animals come in very different forms. One thing to say is that the prion disease are very rare diseases, which affect humans to 1 to 2 individuals per million of population worldwide. But despite this, we have different forms of the disease. The most common form is Creutzfeldt-Jakob disease, but there are other forms of disease, and I'm not going to go into the details of these. Some are sporadic, some are iatrogenic, meaning acquired disease, and some others are genetic. In animal, we have the prototype prion disease, which is scrapie, which affects sheep and goats. And back to 1700 in Great Britain. But then other forms of the disease are quite infamous. There are BSC, or mad cow disease, which was an epidemic which started in Europe at the turn of this century. And most recently, we have another very worrisome epidemic called chronic wasting disease. And this affects most of Northern America. But most recently, we have cases of this disease which affect cervix population, also in Northern Europe. As I said, human prion disease are very rare disease. Here, 1 to 3 cases per million of population worldwide. The most common form is sporadic, 80 to 85%. Genetic is 10 to 15%. And infectious, luckily enough, are only less than 1%. And in this, we can list Kuru, which was due to cannibalism in Papua New Guinea, iatrogenic CJD, and the variant CJD. Iatrogenic CJD is basically due to contaminated surgical instrument or to growth hormone from extracted from human pituitaries. And variant CJD, which is another yet form of iatrogenic infectious prion disease, is due to the consumption of BSE tainted meat products. And this is very important because this was a huge problem worldwide, because this affected quite a bit the health system, because people that were exposed to BAC were not allowed to donate blood, and therefore it put a strain in the health system. And still now, at this day, people like me that spend more than 6 months in Great Britain during the height of this problem cannot donate blood, and therefore you can imagine the problem. So in Great Britain, this was really dramatic epidemics, because it affected the cattle, and as you can see here, the y-axis are with different scales. These are thousands of cases of BSC in cattle, and these are just the number of cases of variant CJD, which is the form of prion disease caused by BSC. So the peak was reached around the beginning of the 90s, and as soon as were put measures to limit this, the spread of the disease, as you can see, there was a very steep diminishing of the overall cases. What worried us was the fact that basically shifted by a few years, there was a surge in the cases of the variant CJD in human, which luckily was basically zeroed after a few years. And we were expecting a higher curve, higher peak of this disease, but luckily enough, this was not. So what are prions? Prions are proteins that, in the basic definition, can adopt at least 2 different. Yes?
Hello, sorry, you're a little muffled right now.
Now it's better?
Yes, it is.
I think I have to lift the computer, otherwise. Okay, I'll lift the computer, no problem. So did you catch this, or I repeat this?
Could you just repeat it so the other people could hear it too?
Okay, so definition of prions. Prions are proteins that can adopt at least 2 different conformations. And here, for simplicity, I draw a circle for the physiological form of the prion protein, and the square is the prion, the pathogenic form of the prion protein. And basically, they multiply by forcing the precursor protein to acquire the second conformation. So as you can see, this, I hope that this is going to be your first take-home message, which define two most important things. First, the prions are proteins, so they don't have nucleic acid to perpetuate their state. And the fact that they can self-replicate by forcing the precursor protein to adopt the pathogenic form. So you would say, well, if this is the central problem of prions, it would be relatively easy to tackle this problem. But basically, there is more than that, because the same precursor protein in mammals, for example, in humans, can adopt yet another or other form of pathogenic conformation. And here I use color coding. So the same precursor protein can adopt different conformation. And the complexity of the problem is even higher, because the same precursor protein can adopt a different conformation, but this different conformation can evolve in a way in the same host or in the same basically tissue by adopting different conformation. So as you can see, it's very, very complex biological problem. So here is what I'm talking about, and here is the left-hand side is what we know being the conformation of the prion protein, which is mostly alpha helical with some short beta sheets and some unstructured part, which is not depicted here. And on the right-hand side, you will see instead one model of the prion, which is outdated now, and I'll show you more recent data on the real conformation of the prion. But basically, you don't need to be a protein expert to see that there are differences between these two objects. And one difference is the fact that the prion adopt a greater part of beta sheets compared to the precursor protein. Can you hear me now? Fine, okay?
Yes, we can.
Okay.
We do have some questions in the chat that
Sure,
we would like to ask you.
Sure.
The first one is, how would you compare the BSE pandemic versus the current pandemic? Are these similar in any ways?
No, well, completely different pandemic, because, well, the BSE was not pandemic, it was an epidemic. So it was restricted to just few countries, and it wasn't worldwide. And the BSE basically was contained the very moment that we realized that the disease, the human counterpart, was due to infected cow with prions. So the moment you stop consuming those products or halting the BSE infection in cattle, then, as you could see from the graph, basically the very moment we adopt measures to halt the spreading within cattle population, the disease disappeared. Here, we are struggling to contain the virus, the SARS-CoV-2 virus, by putting in place measures like wearing masks and not being social and washing hands very often and things like that. But that it kind of very difficult to contain, because when the spreading of the virus is high, you only need a few people that don't follow these rules to spread the virus even further. Now, the BSE is being halted, as you can see from this graph, And I don't see in the short term how the SARS-CoV-2 can be halted unless we develop an effective vaccine. And my prediction, but this is my personal prediction, this will not happen within a year. So if everything goes right with the vaccine development and other restriction measure, this virus will be contained the next fall. But that's, again, it's my personal view, and it's based with the current data. But I mean, there are virologists that think otherwise, that, or even worse, politicians that think even otherwise. So, but I'm not going to enter to that. I hope I answered the question.
Yes. There's another question. Is the precursor form a normal physiological form?
Yes. Yes. This is it's called PrP for prion protein, with apex as C, so cellular form of the prion protein. Instead, the prion is named as PrPSC, capital S, lowercase c, and it stands for prion, and is basically referring to scrapie, so the prototype prion disease. So PrPC is the cellular physiological form of the prion protein. PrPSC is the prion.
Great, thank you so much. That's all the questions we have in the chat for now.
Okay, no problem. So again, we'll come back to the conformation of the prion protein and the prion later on, where I'm going to give you more details on that. So a little bit of molecular biology, but just a little bit. And basically, the prion protein is contained in this PRNP gene in rodents, for example, where we have 3 exons. And in the 3rd exon, there is the complete operating frame of that codes for the protein. The protein is a pre-pro protein, meaning that there are 2 signal sequences. And the mature form of the protein is 209 amino acids. These 209 amino acids contain some peculiar signature peptides called octarepeats, because they are composed by 8 amino acids. And these are basically in the N-terminal moiety of the protein. And this histidine here is capable of coordinating copper. So the protein is a metalloprotein, so can contain copper ions, but other ion as well. But I'm not going to go into this much longer. So it has 2 glycosylation sites at the C-terminus and a single disulfide bridge. So what is the difference between the cellular form of the prion protein, PrPC, and the scrapie form, the prion form of the prion protein? Well, as primary structure, so the sequence of amino acids that constitute the protein, there are no differences. So basically, many studies being performed to see if there are some covalent differences between these 2 proteins, but there are none. The only difference is what I just showed you, is the conformation. And basically, this can be appreciated in the lab when you subject this sample containing either a PrPC or PrPC by limited proteolysis. If you do that with proteases like, I don't know, proteinase K, for example, but other proteases as well, it can digest completely the PrPC, but it cannot digest completely PrPSC. And PrPSC after proteinase K, for example, treatment, harbor a proteinase resistant core of around 140 amino acids, which historically has been defined as PrP2713 because of the electrophoretic mobility of the protein after digestion. So here is a more complete drawing of the prion protein, the cellular form of the prion protein. And here, as you can see, this is what I showed you so far, the carboxy terminal of the protein, which has some structure, mostly alpha helical. But here is the N-terminal moiety of the protein with these octarepeats and histidine residue, which can coordinate basically metal ions. Okay, so again, this is yet another view of the protein where you consider the totality of the different parts of the protein. This is the protein itself. These are the glycosylations branches that are pretty high in compared to the rest of the protein. And this is the protein itself is anchored to the outer leaflet of the cells by a glycosyl phosphatidyl inositol anchor, a GPI anchor. So as you can see, the protein part is roughly 1/3 of the overall protein. What are the functions of the prion protein? Well, for many, many years, we try to define more and more the function of the protein, thinking that the protein itself had a single function. But instead, the protein has pleiotropic different functions that can exert in different cells. The most important ones are the neurite outgrow. The protein itself can serve as a molecular cue for neurogenesis. It can protect synapses and it can regulate the function of synapses. It's essential for maintenance of myelinated axons, especially in the periphery, and in general can serve as protection and survival. So as you can see, he's a protein with very different function in the nervous system. And that's what has puzzled researchers for many years, thinking that, as I said, there was a single function. So how a prion replicates? So this is PrPC in a different form here, where alpha helices are cylinders. And this is PrPSC, yet in this model that I showed you early. The first thing is that these two entities need to get in contact, so to bind to each other. And once this happens, a nascent molecule of prion basically is made. And this cycle can go indefinitely over and over in the nervous system, but not only in the nervous system, but also in other tissues. But basically, it's the accumulation over time of the prions that eventually cause the disease. Here is the most recent scheme of how these prions are made, and basically you can see the cellular form, which is below, that basically acquires a different conformation, and this different conformation is basically all beta now. It is believed to be all beta, so it's not just a part of beta sheets and alpha helices, but the structural conformation that the prion acquires is all beta. And these are even more recent data, structural data done with electron microscope. And basically, these all beta units can pile up one on top of each other and create these fibers, which we know now we define as amyloid fibers, so amyloid deposits in the brain. And this is what we think is one of the possible conformation of a prion. What makes unique prions is the fact that we have very good models. And here I just gather many different experiments done in every corner of the world with the same prion and with the same strain of laboratory mice. And as you can see, despite the fact that you've done the experiment in Australia, in the United States, in Europe, in Asia, whatever, the outcome of the experiment is always the same. So once injected with these, once these mice are injected with this so-called strain of prions, the incubation time, so the time to the disease onset and death of all the animals is pretty much the same. And this is true only for prion disease. Other more common form of neurodegenerative disease like Alzheimer's disease and Parkinson's disease don't have the same robust models. This is what I mentioned earlier, the electrophoretic mobility of the protein. And this is a Western blot, meaning that you first run the protein on by electrophoresis on a gel, and then you blot this separation on paper, and then probe it with specific antibodies. By doing so, and this is after proteinase K digestion, you see that the pattern of the protein is a characteristic 3 bands. And you can see here from different diseases, for example, 2 different forms of sporadic CJD, a form of variant CJD compared to BSC, for example, are very different. And basically, the only thing that you might want to remember from this is the fact that the 3 bands represent the 3 different forms of the protein, the diglycosylated form, monoglycosylated form, and unglycosylated form. Again, prion disease are also known as transmissible spongiform encephalopathies. And here I just want to show you, this is a cerebellum of a mouse injected with prions. This is a control where you don't see much happening in this cerebellum. Another characteristic of prion disease is the formation of these vacuoles in the infected tissues. And these vacuoles are thought to be neurons filled with liquid. And the appearance of the tissue is like a sponge, and therefore here is the name transmissible spongiform encephalopathies. So this is, if you have a question from this first part, which I gave you a very short introduction to prions, I can answer now, otherwise I'll move on to the second part. Do you have questions?
We don't have any at the moment, but please, if you can, drop your questions in the chat so we can see them. Thank you.
Okay. Okay, so basically what we have done in recent years is trying to understand and finding ways of developing new strategies for treatment and eventually cure of the disease. Overall, for all neurodegenerative diseases, we don't have a cure. We have symptomatic treatment, meaning that we can ameliorate the symptoms of the various diseases, but basically we cannot block the neurodegenerative process. So we tried to figure out what was the sets of genes that were upregulated or downregulated upon prion infection. And we started with a model which was in use several years ago, which was the macaque infected with BSE. And by doing this, we can see this microarray analysis, which basically compares the different levels of different transcripts in one group or the other. And by doing this, we found 300 differentially expressed genes, and they basically span over different processes, cell development, lipid homeostasis, and so on and so forth. And interestingly enough, we identify a particular gene, which is called SERPINA3 in the monkeys, which is homologous to the same protein in human, which was very highly upregulated. And this attracted our attention because never before this was reported in the literature. As you can see here, we extended our analysis taking different groups of human diseases, sporadic CJD, GSS, FFI, genetic form of CJD, variant form of CJD, iatrogenic CJD, comparing to Alzheimer's disease. And as you can see, there are some of these prion disease which Serpin fold change is very high, especially for iatrogenic form of the disease, but also for Alzheimer's disease, there was some increase. So this really provide us some basis for further studies. We looked into the protein, and basically the higher the transcript, higher is the presence of the protein. And especially here you can see from the iatrogenic form of the disease. Here is the characteristic 3 bands for PrP, and this is the SERPINA3 protein. And also at immunohistochemistry, you can see there is large deposits of this protein in neurons compared to, for example, Alzheimer's disease, where the deposits are lower. So why and how does prion infection increase in SERPINA3 levels? Here I mentioned SERPINA3N, which is the homologous in rodents. So the event I described you earlier is the fact that we have misfolding of the cellular form of the protein into a misfolded aggregated form of the prion protein. And this can happen for different reasons. There is some amount of these proteins in lysosomes. They cannot be degraded by autophagy. The proteasome cannot deal with this growing amount of misfolded protein, and therefore we have eventually accumulation, which can be, for example, intracellular, in the case intranuclear, for example, in the case of polyQ expansion in certain
Sorry, we can't hear you right now.
Now, can you hear me?
Yes, yes. Sorry.
And basically, some intracytoplasmic inclusion, like Lewy bodies, or extracellular, like in the case of A-beta in Alzheimer's disease. So what we know is that this is the event I was describing before. So the some writers, scientific writers, define this as the kiss of death, when the two proteins come together. And these create the accumulation of prions on the cell surface of neurons. And we have a protease that can, over time, deal with this, lowering the amount. So our current hypothesis with the Serpin, why Serpin is upregulated, is that once prions are formed on the cell surface, these basically start signaling process which triggers the upregulation of certain genes. And among these genes, there is a Serpin A3 in humans or A3N in rodents. These, I haven't mentioned yet what Serpin is, but basically this can block the action of proteases by binding covalently to proteases and then blocking the clearance of prions. Okay, so let's go back one step. So what we think is that prions, and this is another piece of information I haven't told you for the sake of time, is that prion diseases are very long diseases. First exposure to prions can take years before the disease is manifest. So we think over time, the accumulation of prions is dealt by the cells by the use of these proteases, natural proteases, which we think is at the basis of a clearance mechanism that is not yet defined. By upregulating Serpin, these blocks the action of this protease because Serpin is a natural inhibitor of proteases. So it's a protein that itself can block the proteases. Therefore, if we can pharmacologically block Serpin, we could free the proteases from its action and eventually basically lower the prion load. So we did many different experiments in the last few years, and we did some knockout cells for Serpin. And therefore, you can imagine if you knock out Serpin, you free the proteases from its action, so it can digest prions. And we did exactly this. If we, let's go back, okay. Now, this experiment is overexpression of the Serpin. Therefore, if you overexpress the protein, I know it's kind of complex here, but if you knock out Serpin, you free the proteases and you basically chew up all the prions. If you overexpress a Serpin, you increase the prion. And exactly this is exactly what we did with in one experiment where we overexpress Serpin and we increase the prion loads. So let's go to the pharmacological approach. We identify a group of potential inhibitors of Serpin, which could be used as lead compounds for a treatment of prions. Here is what Serpin looks like. And there is this RCL loop, which is responsible for binding to the target protease and make a covalent link. So basically, if we block this RCL to bind to the proteases, basically we should be able to free the proteases and therefore treat the prion load. Here is an expanded view of that. So the RCL loop here, the proteases, the stable binding of the Serpin to the target protease. This is the final covalent complex. And if we block this binding, we can free the Serpin and the protease can be active. One of such complexes very well known is antitrypsin complex, and trypsin is yet another member of this Serpin family. Serpin family is very large, and there are hundreds of different members of this group of protease inhibitors. So by using one of such compound, we call it 5, we could diminish the prion load by 50%. So the compound binds to serpin, serpin no longer is able to bind to the protease. And as you can see here, for example, with this treatment, we can basically abolish the amount of prions present in cells. And we did the titration of all these compounds. And still, we wonder what was the mechanism, because one of the issue here has been, over the years, a lot of the studies have been focusing on binding small molecules to either PrPC or PrPC, and therefore targeting the prion as a primary target for the treatment of prion disease. In this approach that I'm showing you here, we don't go through that. We are binding or targeting a different protein that through a clearance process should deal with prions. And in order to exclude that this molecule we are using are binding to the prion protein, we use this novel test, which is RT-QuIC, basically is quacking inducing the conversion. You put some proteins in a test tube with this fluorescent probe called thioflavin T. By adding shaking, you can basically force the conversion of PrPC to PrPSC. And basically what you can see here from the graph is that if there are no prions present in the sample, the curve, there is no curve, there is a flat line here. Instead, if there is a presence of prions through this amplification, cyclic amplification, you basically have a sigmoid curve that eventually plateau and indicates the presence of prions. Not being the prion protein, the primary target of this compound, nothing should happen. And exactly this is the answer of these experiments. By including the precursor protein and adding the seed of prions or the inhibitors and so on, basically nothing is happening, indicating that most likely this protein, this small molecule cannot bind to the prion protein. So I would like to conclude now, and basically I show you that we identify Serpin as a possible gene involved in prion infection and maintenance. We did some experiments with the knockout cell lines. Now we're working with animals. We'll have this data in the near future. By overexpressing Serpin, of course, you block the protease even further, and then you have a higher amount of prion accumulating. And by inhibiting Serpin A3N, we should be able to reduce prions in cell lines. We're extending this finding in vivo now. So future perspective, as I said, we are moving now to in vivo experiments in Serpin A3N knockout mice. And trying to study in more detail this mechanism of Serpin A3N mediated clearance of prions. Question and answers, yet do Serpin A3N knockout abolish, reduce prion accumulation? We'll see, and of course is our hope. And overexpressing Serpin A3N should provide a model of prion infection which is fast, because instead of taking, as I show you in that graph, 150 days, it could take a week or two to study prion infection. And of course, the work is ongoing for the pharmacological inhibition of SERPIN3N. This, to thank the students that participated to these studies. Here are named few: Arianna, Silvia, Oa, and Marco. And I would like to acknowledge also a collaboration with Marco De Vivo at the Istituto Italiano di Tecnologia, Paolo Carloni at Jülich University in Germany, and Maria Laura Bolognesi at the University of Bologna. And that's basically it. And thank you for your attention.
Thank you so much, Dr. Legname. At this time, we are going to transition into our Q&A portion.
Okay.
Our first
Can
question.
you give me just 1 minute and I'll be back? Just 1 minute?
Yeah, definitely.
Okay. Thank you.
Students, this is a great time for you guys to reflect on the presentation that was just given by Dr. Legname and think of any questions that you may have. Feel free to drop them in the chat. This doesn't have to pertain only to his research, but you can ask him about his background, how he got into research, his day in the life as a researcher, etc. If you're curious about anything that relates to not only how he got there, but how he continues his life as a researcher and what that entails on a daily basis. So we're going to start with our questions. Rafael is going to go ahead and ask you the first question.
Sure.
There was a question regarding when you said kiss of death, why is it called this? Why is it called the kiss of death?
Sorry, can you say it again?
What did you say the kiss of death was, and why is it called?
Okay. The kiss, I mean, I just mentioned this because it was a popular definition that was given by an author. Now I don't even remember the name of the guy. Basically, the binding of PrPC to PrPSC was defined as the kiss of death. The kiss of death. I mean, as soon as these two proteins come to contact, then you're doomed because the PrPC turns into PrPSC.
Thank you. Is there any particular reason the pathogenic form of the prion proteins assume the beta sheet shape?
Because yes, because this is energetically is favored. Protein with the high content in beta sheet are more stable because they require lower energy to stabilize their conformation, while it's more difficult to keep together a protein with other secondary structure like alpha helices. So this is the reason why, I mean, there is a famous plot where you can look at the protein stability and you see basically it's like two valleys and a peak of a mountain, where the two valleys, one is higher and the other is lower. And the higher is the cellular form of the prion protein. If you can overcome the energy barrier, the beta sheet allows the protein to get a more stable conformation into a beta sheet.
Another question is protease X capable of degrading PrPSC?
Yeah. Well, you know, all protein can aggregate. Under certain condition, you can misfold the protein, and the protein will search for a stable conformation. Most of the time, as I said, without a cellular machinery that helps, like chaperones or other environmental condition that helps the protein to assume the native conformation, most likely when you misfold the protein and then you try to fold it again in vitro, it will aggregate. It will aggregate and find itself in a very high content of beta sheet. The classical example is when you cook an egg. I mean, when you cook an egg, basically you have the protein of the yolk and of the egg. When you inject energy into the system, then it solidifies, and that's not amyloid, but basically the protein misfold and aggregates and solidify.
The next question is asking about your educational background. So which university did you go to?
Okay, so my career was at the beginning very erratic because I studied my bachelor degree and master degree in Milan. And I was lucky enough to get a job in a pharmaceutical company back then, right after my master degree. And I went to Great Britain to work, and there I decided to do a PhD course. And luckily I did this because I didn't like to pursue my career in the pharmaceutical industry, and I decided to go back into academia. And with my PhD, I found a job first in London at the Medical Research Council. Then I came to California for 8 years to UCSF. And then eventually I moved back to Italy, where I work now. So it was not a straight path, but
What inspired you to go into research?
curiosity. You know, you have this urge to understanding the world around you better. And you know, there's one thing that for those that do research, we consider ourselves very lucky because we do a job that we like. So we try to solve puzzles and so on. Before the pandemic, we traveled a lot, and so we know other environments, other setting, and we meet a lot of interesting people. So in this respect, we're really lucky. But I think mostly is curiosity, trying to understanding our world better.
Thank you. Going off of that, why did you decide to research prions?
Okay, so that was just break of luck, because I was working in immunology in London, and I was pretty happy there with my family and so on and so forth. And I received a phone call from Stanley Prusiner, who's the one that defined prions, the prion field. And he asked me to join his team in San Francisco. And of course, if you compare the weather between London and San Francisco, I prefer much better the weather in San Francisco. No, I'm joking. No, it's not because of that. One of the things I learned to do during my early stage of career was to make recombinant proteins. And Stan back then needed somebody to help him produce recombinant protein. Therefore, I decide, which many colleagues of mine at the time told me that I was crazy because I was doing well in immunology, but I decided to switch completely career and then go into prion research.
The next question is, how does US research compare to researching in other countries?
It's completely different. I mean, I do remember that working in the United States is much, much easier. And of course, if you have funding and if you're working in a good institution and things like that, you anything that you need for your research is at your disposal right away. In other countries, this is less so, and I know I can compare Europe and US. In Europe, it's a little bit more difficult because some of the reagents we use, they actually come from the US. So it takes longer for us to get reagents there. But I think US has a very good system in supporting researchers and supporting people that want to go into this job. And Europe is the same. The pace of research is a little bit slower in Europe than in US. But other than that, the science is science.
Thank you. Have there been any negative outcomes observed in inhibition of Serpin A3?
That's a very good question. We are learning now. We are learning because apparently the phenotype of mice lacking Serpin is normal. So we cannot say that lacking Serpin A3N in mice could lead to certain pathologies. But because the system there, again, I probably mentioned somewhere in my presentation, there is no one Serpin. Like there is no one proteases, target proteases. So Serpin family is very large. There are many members and proteases as well. So if you get rid of one inhibitor or one proteases, nothing really changes, or at least as far as we can tell, it doesn't really disrupt the physiology of the cell. But we are learning this. So we are actually studying more carefully the knockout and see what happens.
And then like next question off of that, does Serpin A3 only target infected prions for degradation?
Okay, so this is another good question. So basically, what we are doing now is trying to identify the proteases. Because despite the fact that the Serpins are all very similar to each other, our thinking is that one Serpin may block one proteases and may not be blocking other proteases as well. So we are doing now real-time PCR to identify which protease is the target of Serpin A3N. And we think we identify one member. So basically, only one protease can be blocked by Serpin. And the proteases, usually, these extracellular proteases I'm talking about are the one that serve the tissue to remodel itself. So they are there for clearing any, let's say, unwanted protein or protein complex for the growth of the tissue or the cell. So we think that this may be the mechanism, but we are really studying this still.
Thank you. Are there currently any promising potential drugs for blocking the RCL loop from binding and inhibiting the protease?
Yes.
Or human trials down the line?
No, not yet, because the molecule we found, the molecule that I showed you earlier, it's very good in vitro, but in vivo doesn't cross the brain-blood barrier. So we need to extend our finding in identifying new molecules that are capable of crossing brain-blood barrier and still maintain the inhibitory activity with Serpinan. This is our hope. This is our hope. If this mechanism that I show you holds true, then developing one such inhibitor could really change the way we approach the treatment of the prion disease. But as I show you, could also be true for Alzheimer's disease and other neurodegenerative diseases.
Thank you. Can you talk about how your research has been affected by the COVID-19 pandemic? Like, how did you have to adjust your research and stuff like that?
It's been a disaster. In Italy, it's been a disaster because basically in March, when we had the lockdown for 3 to 4 months, we couldn't really go to work. We stayed home. So my students struggled to keep up with their research, but they couldn't do experiments. So we actually decided as a school to give an extension for their PhD in order to make sure that they can do all their experiments. After the lockdown, there was a slow coming back to day-by-day life. And I have to say, I've been working from home ever since the first lockdown, while the students and the other researchers have been, with all the precaution, of course, back to the lab to do experiments. But it's been difficult, I have to say, very difficult, and still is, because I guess our Prime Minister tomorrow will announce new measures because there is a surge in cases of COVID-19, and therefore we will go to another lockdown, who's not going to be as tough as before, but still, I don't know how this second wave can affect our work.
Thank you. Our next question is, how do you recommend students get into research during the pandemic?
If I recommend somebody to go to research?
No, how do you recommend them pursuing research during the pandemic? Like, how do they find,
the one thing that really worries me a lot is that a lot of researchers nowadays are moving towards studying the SARS-CoV-2 right now, and COVID-19 and so on and so forth. And this is worrisome because not everybody is equipped or has the basic knowledge to actually attack this problem. I mean, a virologist need a training that will require several years in order to do that. And I see that people coming from different environment or background moving towards this research because there is funding and people need funding to do research. But I would be very careful and cautious in doing so. For example, I decided ever since the beginning not to do so because, again, I think there are people more prepared than me to actually study this virus and carry on doing what I know to do best. Because once this pandemic disappears or is controlled, we still have to deal with the neurodegenerative diseases. We still have this, as I show you at the beginning, we still have to treat people, or at least find treatment for these people for such devastating diseases. And I wouldn't do my job at its best if I start jumping boat from one field to another just because there is funding. On the other hand, I can see that people that are good virologists are actually shifting their research into only this. And of course, this is very, I would say, useful for all of us because we still to understand better this virus.
Thank you. The next question is, what qualities / characteristics or skills do you look for in assistants in the lab?
Well, first of all, it needs to be a person that is highly motivated in what he does. I think every researchers, every PI needs a very good lab assistants and eventually lab managers, because they are the one that actually hold the boat on a daily basis. And basically, I think what I look for in people like this is how well they organize their work, how rigorous they are in doing the experiments. Therefore, even if there is a negative result or a result that cannot be explained at first, to be critical and defend their work as long as they believe that they have done nothing wrong. So one thing that is very important in science is being rigorous and honest. There are no shortcuts. There are no shortcuts because science always wins. If you trying to push some data because you want to publish well and so forth, or others, eventually, if there are not real data, you will be, other people find out. So, I mean, you need to be sure that whatever you're publishing, whatever you can defend this at the very end, because you know that these data are true. So honesty and being rigorous with one's work is, I think, is very important.
The next question is, can you talk about what your day-to-day life is like doing research pre-pandemic and then post-pandemic?
Well, pre-pandemic was the typical day was, when I was not traveling, was to go to work, to my office. Unfortunately, I'm not doing experiments any longer, and meeting with the students, postdocs, lab assistants, and so on, to organize and check how the various experiments were going. Lots of meetings, also for with faculty, for the school, and so on and so forth. And of course, the most enjoyable part of the day was writing a paper or organizing somebody else work, like a student work in their thesis and so on. During the pandemic, I stay home most of the time, and I've, I think, published close to 20 papers this year, because I was doing only that. I was only writing and organizing paper, which is very nice for all my students and postdocs, because finally they can see the light of their work. But I mean, I'm enjoying myself despite this problem. But I hope this will finish soon.
Going off of that, do you have any recommendations for people who want to publish their own work or publishing under someone?
Well, first of all, it's very competitive. Our job is becoming extremely competitive. I mean, when I started last century to do this job, it was relatively easier, because you could publish a nice paper with one piece of data. Now, to have a good publication, you need lots of data, look at the problem from different angles, try to find more and more evidence to support your thesis, and so on and so forth. But well, you know, publishing depends a lot on the PI, of course, because he holds the strings of the funding and the responsibility at the end of what has been published. But if you have a good PI, if you have good data, I don't see what would be the problem.
Do you have any recommendation on how to find like a good journal to publish your work in?
Well, you know, nowadays there is all these ranking of journals for impact factor and so on and so forth. My suggestion is not to look at impact factor. And if your research is good and solid, any journal is, any good journal is fine. I just want to remind the people that don't know, whoever invented the PCR, polymerase chain reaction, he was rejected by Nature, and he had to publish the paper in a lower so-called ranking journal. But then the guy then won the Nobel Prize. I mean, it doesn't really matter. I mean, of course, especially in the US, but I think worldwide, of course, publishing well, high impact factor journals and so on allow you to have a better career. But it's not always true. I mean, again, if you are a good hard worker, publishing well and good solid piece of data, it's enough. Because eventually, if what you publish holds the time over and over again, it's only, that's all that matters. That's what I said before: honesty and integrity and rigorosity. I mean, that's what eventually pays off.
Has there any been permanent changes in the germline for those who had prion disease?
That's a good question. Well, you know, one thing that puzzles us still is the sporadic form of the disease. There's been a paper just published where they try to see if there are modulators of gene that modulate prion disease. And of course, besides the prion gene, there were one or two other genes that were identified. And but we don't know. I mean, the sporadic cases are still a puzzle to us. They're not being linked to any other gene or modification or mutation of other genes. And therefore, we don't know what's going on there. I mean, we still need to look for something. There is an interesting idea that some sporadic forms of the disease may be due to somatic mutation. I mean, mutation that occur after germline establishment, but still this is the early stages of research. So, but it's intriguing that some genes can mutate later once a germline has been established, when a cell has been established, and maybe this could trigger the neuropathological mechanism, but we don't know.
I'm sorry, do you know if it can be passed down to offsprings?
Yes, I mean, the genetic form, yes. But the penetrance is not 100%. There are carriers with the mutation, they may be healthy throughout their life, while others, they are not so fortunate, they have a higher penetrance. So the genetic form of the disease, yes, they can be passed to the offsprings. And of course, only one of the two parents has passed through the affected gene to the offspring.
Thank you. Do you recommend students get into research during their
Absolutely,
undergrad?
absolutely. If you're curious and one thing that you have to understand is that doing science is like playing. You want to build something with Lego. You want to. I still remember how excited I was when I cloned the first gene and I sequenced the first piece of DNA. And I couldn't sleep because I wanted to have the answer the next morning. I mean, if you are interested in getting excited on a daily basis on something like this, yes, go to research. If you don't want to do that, you rather do some other jobs, that's fine too. That's fine. But definitely research for me, especially, has been an exciting journey so far.
Thank you for that. My next question is, sorry, hold on.
It's okay.
What qualities, characteristics, or skills do you look for in students applying to work with you?
Academic track record is the first thing, because people need to have a basis knowledge, background on what they want to do. Enthusiasm is another thing that you when you see this spark in the eyes of people when they talk about science, and that's always very welcome, meaning that people are really excited on this. And I think overall it's perspective. I mean, how, but this is, cannot be quantified. I mean, you need to look at a person by talking to them and see how you think they could be successful in this job.
Great, thank you so much. Our final thing that we want to ask you is if you have any last minute advice for our students.
Study a lot and do whatever you like, because there is no better things to spend your life doing whatever you like. I think this is the most important thing. I mean, you shouldn't be forced to do things that you don't like. If you want to do research, if you want to become a scientist, study a lot, because studying is never enough. And just go into something that you really enjoy. You won't feel you won't feel the pain and the which this job carries, because the bad experiment or get rejected by a big journal or things like that. These are things that happens all the time. So you just need to keep going and forget about this.
Thank you so much for this educational and insightful speech. Students, please, can you stay until the end of the session? Nina will talk about how to take the post-shadowing assessment. And if you live in the United States, please, please, please go out and vote if you haven't, especially for those who can't. Thank you.
I concur. Okay, thank you very much for hosting me. If you have any follow-up question, just Nina, just write to me. I will be happy to answer that. And
Great. Thank you so much. I want to go ahead and tell my students, if you guys want to tell us about anything that you heard today in this presentation that particularly resonated with you, anything that you learned, any big takeaways, please leave a comment under our speaker's profile on our website. This will be at the very bottom. You will see a little comment section, and you guys can let us know how you found this talk. It's always very appreciated. If you are interested in joining the team, we do have applications open. You can find this on our website. And if you do not have enough time to commit, there's no worries. We also have volunteer opportunities, which allow you to work from home during the pandemic for as much or as little time as you want and on your own schedule. So if you can only fit in 30 minutes before you wake up or 30 minutes before you go to bed, that is completely fine. You can do it whenever you can.
Okay.
Our next announcement is to go to our Instagram page. Our Instagram, you can find it, is @PreHealthShadowing. And we have a challenge for you today. If you go to our speaker's post on our page and you type out his last name letter by letter, you will be eligible to be picked for the student of the session. And whoever can get his name all the way down without getting interrupted by another student can work. Yes, the volunteer page is available on the website. If you go to www.prehealthshadowing.com/volunteer, you'll be able to find it. Our next shadowing session is Tuesday, November 10th at 10:00 a.m. Pacific Standard Time. We will be hosting Myra Tibet, who is a veterinary doctor. And thank you again for coming to our shadowing session. If you would like to have your certificate, which verifies your hours today, you must take the post-shadowing assessment. To do that, you want to go to the website, www.prehealthshadowing.com, and click Take This Quiz. Once you are in the quiz, you will have 15 minutes to complete it. Please be sure that you click Next rather than Complete when answering each question. When you click Complete, your quiz will be turned in, and if you have not finished, you will fail. You will have one retake to successfully pass with a 70% or higher. Once you pass, you have to go back to the speaker's course page and click Finish Course. If you do not click Finish Course, you won't receive your certificate. Please make sure to click Finish Course. Once you do that, which can be downloaded immediately or can always be found in your profile. If you guys have any questions or experience any issues, please feel free to reach out to us at www.prehealthshadowing. Nope, not www, it's an email, info@prehealthshadowing.com. Again, that's info@prehealthshadowing.com. If you guys have any inquiries, questions, concerns, we got you and we will respond to you as soon as we can. That concludes our 11th session. Thank you all again for joining, and we look forward to seeing you at our next one.